Bismillah, assalamu alaikum. We are going to talk about the myelinated fiber. What we mean by myelinated fiber, that means this axon is completely surrounded by a fatty material, and this fatty material contains phospholipid, cholesterol, and protein. And this lipid-rich material is form an insulation for the axon for protection and to increase the velocity of the conduction of impulses. As we can see here in this diagram, H and E protein stain. In the middle we can see the axon in red. White area which surrounds the axon is the myelin sheath. Axon white area represents the myelin sheath, and this myelin sheath white appears white because usually during the preparation of the H and E stain it dissolved, the lipid material dissolved, so it appears as a white area surrounding the axon. For that we use another type of staining which is called the osmium fixation or the silver stain. We use sometimes silver stain to stain the lipid, and this lipid usually, as we can see, it appears within the osmium fixation as a dark brown area which surrounds the axon. So the axon in the middle white, and this area appears as the myelin sheath which surrounds the axons. Usually myelin is formed by Schwann cells within the peripheral nervous system, and within the central nervous system it is formed by the oligodendrocytes. The two cells are responsible for the formation of the myelin within the peripheral nervous system. It is the Schwann cell and the oligodendrocytes within the central nervous system. How the myelin is formed? Let's see. As we can see, the myelin here, each myelin forms an internodal segment. This is the internodal segment within the peripheral nerve which surrounds the axon. Each one cell represents an internodal segment which wraps around the axon, and each internodal segment is surrounded by the node of Ranvier. This is the node of Ranvier. Node of Ranvier does not have any myelin at their side, and usually they are rich of mitochondria and they are the side of the collateral axons. Usually the myelin is the Schwann cell engulfs the axon, invagination within the membrane of the Schwann cell, and then the Schwann cell start to surround the axon. Initially the membrane of the Schwann cell become opposing to each other, and this is called the mesaxon. Then the mesaxon and the membrane will continue to wrap around the axon many times in a concentric layers of membrane, as we can see, until the axon is completely surrounded by the membrane of the Schwann cell. At this stage it is called myelinated axon. So this is the nucleus of the Schwann cell, this is the myelin, the axon, and this is the inner mesaxon, the meeting of the opposing membrane, and the outer mesaxon. This is how the myelin takes place. In longitudinal section, as we can see, those are the layers of the membrane. This is the nucleus of the Schwann cell, and this is the axon and the membrane of the Schwann cell which wraps the axon many times, and this is the node of Ranvier. This is the node of Ranvier. Nucleus the membrane of the Schwann cell. In certain locations, the membrane of the Schwann cell is not completely opposing to each other, not completely met. Some cytoplasm left in between the membrane. So those are called the Schmidt of Lantermann clefts. Usually those clefts are arrow shape, usually, as we can see here, arrow shape. Again, Schmidt of Lantermann representing arrow shape structure, which in fact not completely opposing between the membranes of the Schwann cells, the membrane of the Schwann cell itself. Again, this is the node of Ranvier, no myelin located in the node of Ranvier. Again, this is the node of Ranvier, the membrane of the Schwann cell cytoplasm, and this is the node of Ranvier under the electron microscope. No myelin. Within the node of Ranvier, we can see lots of mitochondria. And why? Because it is the site of saltatory reaction, or the jumping of the impulses, and a site of the collateral axons. There are certain axons within the nervous system which are non-myelinated fiber, which means they are not surrounded completely by a membrane or concentric layers of the membrane of the Schwann or the oligodendrocytes, which forms the myelin sheath. And usually those non-myelinated fibers located at the post-ganglionic fibers of the autonomic nervous system. Those are examples of where we can find the non-myelinated fibers. What we mean by non-myelinated fibers, we can see in those diagrams. In the first one, in this diagram, we can see this is the nucleus of the Schwann cell. Within the membrane, we have clefts, and those clefts contains the axons, or in certain locations we have many axons within those clefts. In both diagrams, those are representing a non-myelinated fibers. In the non-myelinated fibers, we can see there are no multi-layers of the membrane which surrounds the axons. Instead, only the Schwann cell engulfs the axons without wrapping around it into many layers. And in those clefts, we can see a single axon or many axons surrounded by this Schwann cell, and this is the nucleus of the Schwann cell. So within the non-myelinated fibers, still we have Schwann cells. Instead, these Schwann cells, they do not have many layers or membranes which surrounds the axons, and those types of axons are called non-myelinated fibers. Usually, non-myelinated fibers are smaller in diameter than the myelinated fibers, and usually they conduct impulses in a lower speed than the myelinated. This is the myelinated fiber, myelinated axon, many layers of the Schwann cell membrane surrounding the axon, and this is the nucleus of the Schwann cell. On the other, this diagram, this is the non-myelinated fibers. Only clefts which can surround axon or multiple or group of axons. Again, this is how the myelin sheath form. Initially engulfing mesaxon, start to wrap into many layers around the axon until the axon is completely myelinated, while in this case this is a non-myelinated fiber. Still, in both we have myelin sheath. The difference is that the myelin sheath is not surrounding the axon within the non-myelinated fiber in a form of multi-layering. This is an electron microscope image with color gradients. As we can see in this section, this is a myelinated axon, transverse section with myelinated axon. This is the nucleus of the Schwann cell, and this is the myelin sheath which surrounds the axon. This is the axon, myelin sheath, and nucleus of the Schwann cell. On this second diagram, also it is a scanning image, transmission image of within a transverse section with color gradient. We have non-myelinated fiber. This is the nucleus of the Schwann cell, and those are the axons which are engulfed within the membrane of the Schwann cells. As any part within our body, each tissue has its own innervation of a blood supply. The connective tissue within the peripheral nerves usually surrounding a whole nerve trunk, and the connective tissue which surrounds the whole nerve trunk or the whole peripheral nerve is called the epineurium. This is the epineurium. It is the connective tissue which contains blood supply which surrounds the whole nerve trunk or the whole peripheral nerve or the nerve bundle. The group of axons which are surrounded by a connective tissue, this connective tissue which surrounds a fascicle or a group of axons is called the perineurium. And finally we have the endoneurium. The endoneurium, the connective tissue which surrounds an individual fiber. As we can see, this is the connective tissue which surrounds the myelinated axon. So we have endoneurium for a single axon, perineurium for a fascicle or a group of axons, and the epineurium which surrounds the whole nerve bundle or the whole peripheral nerve. Those are light microscopy of peripheral nerve or nerve trunk. As we can see, those in the 4 slides, those are peripheral nerves. In those 2 sections, there are stains with H and E, while in those sections we use the osmium fixation, or sometimes we use the silver stain. As we can see, those small rounded structure, each one of those representing the axon which is surrounded by the myelin sheath. We said that the myelin sheath usually in H and E appears as a white area, and in the middle of this white area we have the axons. This is the epineurium which surrounds the whole nerve trunk, and the group of nerves or axons are surrounded by the perineurium. The individual fiber is surrounded by the endoneurium. Again, H and E stain within a peripheral nerve. Those are the axons, and usually axons are surrounded by the myelin sheath. Endoneurium surrounds the individual fiber. Perineurium, connective tissue which surrounds the fascicle, and the epineurium surrounds the whole nerve trunk. In this is the osmium fixation. We used osmium fixation to stain the myelin, the lipids which surrounds the axon. So darkly stained area, brown to black areas, represents the myelin sheath. Those are number of axons. Each rounded structure of those presents a transverse section of an axon. Perineurium surrounds a group or fascicle of axons, while the endoneurium, it surrounds the individual axon, while the epineurium surrounds the whole nerve trunk. At higher magnification, we can see this is the axon in white area. This is osmium fixation axon surrounded by this dark band representing the myelin sheath. The endoneurium represents the connective tissue which surrounds the individual axon. Perineurium, which surrounds a group or fascicle of axons. Those are perineurium, and this is another perineurium, while the epineurium, which surrounds the whole nerve trunk. Again, this section representing a peripheral nervous system with osmium fixation. Now we move into another subject which is called the axonal transport system. What we mean by axonal transport system? The axon or the neurons in general, as we said, they are highly active cells. So those cells need to synthesize their substances within their own cell body. And as we said, some axons are short, others are long. Within those axons or within those neurons, there should be a way of transporting the substances or the chemicals across the neuron. And usually those chemicals or substances are transported via what we call the axonal transport system. This axonal transport system composed of neurotubules and neurofilaments, mainly the neurotubules, which are microtubules. This axonal transport system is bidirectional, which means it goes in both directions. First one is called anterograde. What we mean by anterograde, it is the transport of substances from the cell body to the periphery or towards the cellular processes. On the other hand, the retrograde, it is from the periphery towards the cell body or the pericardium. So the retrograde is the transport from the axon and the dendrites towards the pericardium. And usually within the retrograde, this time the toxins and viruses transported towards the pericardium, particularly within the polio. Usually polio virus, which causes the poliomyelitis, it is transported from the ventral columns of the spinal cord towards the ventral column and cause paralysis. This is for the motor neurons. While within the sensory neurons we have the syphilis, which usually transmitted towards the dorsal columns of the spinal cord. again we have anterograde transport towards the periphery, while the retrograde is the transport from periphery towards the soma or the pericardium. On the other hand, this axonal transport system, either it is a slow or fast, slow axonal transport system or fast axonal transport system, and this depends on the conduction of velocity of the transport across the neurotubules. The slow axonal transport system, it is a single direction system, which means it is from the cell body towards the periphery or the terminals of the neurons. For that, it is only anterograde. So the slow axonal transport system is only anterograde, which means from the cell body towards the periphery. Why it is slow? Because it conducts or it transports the substances in a speed of 0.2 to 4 millimeter per day. The fast axonal transport system, usually it transports substances in a speed of 20 to 400 millimeter per day, so it is of higher velocity than the slow axonal transport system. This fast axonal transport system is a bidirectional, which means it can occur in anterograde and in a retrograde manner. The slow axonal transport system usually transport the components which are needed for growth and regeneration of the axons, while the fast axonal transport system usually transport substances which are involved in the transport of enzymes and neurotransmitters towards the terminal or towards the synapses. So we have anterograde and retrograde. We have slow axonal transport system, which is only anterograde, and we have fast axonal transport system, which is anterograde and retrograde, which acts in both directions. Within the central nervous system, we have different types of supporting cells. وحكينا supporting cells وبالإضافة للنيورونز, they are originally from the neuroectoderm. The neural tube, lining of the neural tubes makes differentiation for all types of the cells of the nervous system, which includes the neurons, the major structural and functional units, and the supporting cells. What are the functions of those supporting cells? Their function includes physical support. They can protect the neurons and their processes. They provide electrical insulation for the cell body and the processes as well. They can provide metabolic exchange between the neuron cells and the processes and the blood vessels, and they can participate in the blood-brain barrier. Also, they participate in the repair process following damage or injury of the neurons. Those supporting cells are non-conducting cells, which means they cannot conduct impulses. The only cells within the nervous system that could conduct impulses are the neurons. So there are certain differences between the neurons and those supporting cells. Those supporting cells have no action potential, which means they do not have conduction or propagation of impulses. they are non-conducting cells. Also supporting cells, they are able to divide, while the neurons, they do not have the ability to divide. As we recall, they are called post-mitotic cells. The neurons, they are intended to last for the whole of our life, while those supporting cells, they can divide, and those supporting cells are the site of the tumors within the nervous system. There are no tumors for or originated from the neuron. As we said, the neurons can't divide. Supporting cells, they do not form synapses, unlike the neurons where we can find many synapses in between them. In fact, within one neuron we can find many synapses. Supporting cells, they are non-myelinated, unlike the axons of the neuron, which are usually myelinated. Those are generally the differences between the neurons and the supporting cells. Those supporting cells حكيناهم through our lectures. They are divided into peripheral supporting cell and central, the peripheral nervous system supporting cells, and the peripheral and the central nervous system. So they are divided into peripheral nervous system and into the cells which are present within the peripheral nervous systems, and the cells which are present within the central nervous system. Within the peripheral nervous systems, or the supporting cells of the peripheral nervous system, we have two types of cells, only two types: the Schwann cells and the satellite cells. Schwann cells, or which is called the neuroliminal cells. Neuroliminal cells. So the Schwann cells, they are called the neuroliminal cells. Main function of those cells is to form the myelin sheath. Okay, each Schwann cell, they form an internodal segment. In between those internodal segments, we have the node of Ranvier, where there is no myelination. Also, the myelination is absent from the dendrites, the axon hillock, node of Ranvier, and the terminal ends or within the synapses. Those are the major locations where we do not have myelin sheath within and myelinated fibers, or within myelinated neurons. The second type of cells are called the satellite cells. Usually satellite cells, they surround the pericardium or the soma. They are cuboidal cells, and those cuboidal cells, they provide electrical insulation and metabolic exchange between those cells and the blood vessels, and they do not have the which we can find within the peripheral nervous system. The Schwann cell, which forms the myelin, and those are the satellite cells. Usually satellite cells, they are associated within the pericardium or the cell body of the neuron. This is the nucleus of the neuron. In a cross section, we can see those are the satellite cells. They provide metabolic exchange and electrical insulation for the neuronal cell body. In general, they are cuboidal in shape. Those are the satellite cells which surrounds the neuron, and this is the pericardium or the neuron. This is the nucleus of the neuron. Look at the difference, as we can notice the difference in size between those supporting cells and the neuron. The neurons are very huge cells compared to those supporting cells. So, satellite cells and Schwann cells. Schwann cells form the internodal segment within the axons, while the satellite cell surrounds the perikaryon or the soma of the cell body. Now we move into the supporting cell of the central nervous system. They are called the neuroglial cells, or the glial cells, neuroglia, or the glial cells. They are the supporting cells of the central nervous system. Within the central nervous system, we have four basic types of supporting cells, or we have four neuroglial cells, and those include astrocytes, oligodendrocytes, microglia, and ependymal cells. The astrocytes or the astroglia and the oligodendrocytes, which is called also oligodendroglia, they are macroglia, which means they are large cells compared to the other two types. So the astrocytes, oligodendrocytes are macroglial cells, while the microglia and the ependymal cells are small cells. The supporting cells of the central nervous system, they form 40 to 50% of the total volume of the nervous system. Let's start with the astrocyte or the astroglial cells. Those are the most abundant types of cells within the central nervous system. As their name indicates, astra means, or aster means star shape. They are star shaped cells. They contain long cellular processes with a star shape, and some of those star processes which form the star shape, they form perivascular foot process, and those perivascular foot process usually they contribute in the blood-brain barrier. They communicate directly with the blood. One of those functions they have, those astrocytes, they are the most important because they have many functions. They can provide metabolic and physical support for the central nervous system neurons. Also, they have a maintenance function. They can maintain the composition of the extracellular fluid within the central nervous system. They can remove transmitter from synapses. Also, they are called the scar-forming cells within the central nervous system. So they are the most important of all supporting cells within nervous system. Also, they are the most abundant, which means we have lots or high number of astrocytes compared to the other types of the supporting cells within the central nervous system. Those astrocytes, they are protoplasmic astrocytes and fibrous astrocytes. Protoplasmic astrocytes, they are called mossy cells. What's the difference between those types? This diagram representing the astrocytes, star-shaped cells. It has a foot process and they can directly communicate with the blood to maintain the blood-brain barrier, and they can control the extracellular fluid. Those are the astrocytes with their perivascular foot processes, while those present within the gray. To distinguish between the protoplasmic astrocytes and the fibrous astrocyte, as we can see in this diagram, this is the protoplasmic astrocyte. Protoplasmic astrocytes, usually it has the thick cellular processes, and those cellular processes are highly branched. The cell body of those cells are rounded cell body, nucleus oval to rounded nucleus, almost the cell body completely rounded, and as we can see, they connects with the blood vessels with their perivascular foot processes. Protoplasmic astrocytes usually only present within the gray matter. Unlike the fibrous astrocytes, this is the fibrous astrocytes. We can see they have thin cellular processes, not so branches like the protoplasmic. Cell body as polymorphic, which means the changing cell body, not completely rounded cell body with oval nucleus, and even those cellular processes are very thin, no branches of those cellular processes, and usually those cellular processes very thin, as we said, and this fibrous astrocytes only present within the white matter, unlike the protoplasmic astrocytes, which are present within the gray matter only. Functions and types of the astrocytes. In this micrograph, we can see those are the astrocytes, as we can see. Star shape with many processes, and this section is from the gray matter, so those cells are protoplasmic astrocytes. This one from the white matter, and this represents the fibrous astrocytes. Again, this is an astrocytes. This is the cell body, pericardium, and those are the processes of the astrocytes. This is an immunostain of the astrocyte. The second type of the supporting cells within the central nervous system is the oligodendrocytes. Oligodendrocytes or oligoglial cells. Usually, those cells also contains processes, cellular processes, but those processes are fewer and shorter than the astrocytes. The main function of those cells, they are resembling or analog for the Schwann cell. So they can form the myelin sheath, but those within the central nervous system. The Schwann cell, it forms myelin within the peripheral nervous system. So they are homologs to the peripheral Schwann cell. The only difference between the Schwann cell and the oligodendrocytes, beside the location, we said that the oligodendrocyte within the central nervous system, while the Schwann within the peripheral nervous system. The other difference is represented in this diagram. As we can see, this is the oligodendrocytes. The oligodendrocytes can send their cellular processes to wrap the axons within the central nervous system, which means each oligodendrocyte can form many axons, many myelin sheath around many axons through their cellular processes. while within the peripheral nervous system, the Schwann cells can only form only one internodal segment. So this is the major difference between the oligodendrocytes and the Schwann cell. Both the Schwann cell and the oligodendrocytes, they can form myelin sheath. The Schwann cell can form myelin sheath within the peripheral nervous system, while the oligodendrocytes can form myelin sheath within the central nervous system. Oligodendrocytes can send their processes to form the myelin sheath around many axons via their processes. بينما Schwann cell within the peripheral nervous system can only form a single internodal segment. This is under the microscope. This is an oligodendrocyte which forms the myelin within the central nervous system. The third type is the microglial cells. Those cells are small cells, as we can see. Micro means small. Small cells. Also, they contain cellular processes, very branched processes, and they can found within the white and the gray matter, in both the white and the gray matter. And those cells, they are the neural macrophages, which means they can phagocyte any damaged particle or any foreign substances or particle within the central nervous system. Some textbook says that they are of mesenchymal origin, not ectodermal origin from the neural tube like the other types of neuron nervous system cells. Those cells, they are originated from mesenchymal origin, particularly from the monocytes, because they have macrophage or phagocytic ability. Although still some textbooks says that they are of those cells are originated from the neural tube. The final type of cells or supporting cells within the central nervous system are the ependymal cells. What we mean by ependymal cells? Ependymal cells, they are the lining cells, epithelial lining. They form the epithelial lining within the cavities within the central nervous system. They form the ependyma or epithelia within the central nervous system. Usually we have within the spinal cord the central canal. This is the central canal, and this canal usually lined by ependymal cells. Also within the brain we have cavities which are called the ventricles, and those ventricles also they are lined by ependymal cells. The morphology of those ependymal cells, they are cuboidal to low columnar cells with cilia, ciliated columnar cells. They line the cavities within the central nervous system. Those ependymal cells, they are usually backed by the cerebrospinal fluid. Cerebrospinal fluid is the fluid which surrounds the central nervous system all through. And this cerebrospinal fluid, in fact, it is produced by a modified type of ependymal cells with associated capillaries. Both ependymal cells and some types of capillaries, they can produce the cerebrospinal fluid. And those structures Both they are called the choroid plexus of the brain ventricles. So the choroid plexus, which produces the cerebrospinal fluid, it represents ependymal cells and sometimes of capillaries. Again, the arcuboidal to locolaminar ciliated cells, they lines the cavities within the central nervous system. As we can see in this diagram, we can see the whole neuroglial cells with the neuron. Those are the neurons, as we can see, the largest cell of all, and those are the oligodendrocytes, which sense their processes to form the myelin around the axons of the neuron. And those are the protoplasmic astrocytes, as we can see, and usually those present within the gray matter. Star-shaped cells with long cellular processes, and those small cells, usually fibrous astrocytes, which usually then present within the white matter. And those are the microglial cells, as we can see, they are the macrophages, and those are the ependymal cells, the lining cells of the ventricles and central canal, or the cavities of the spinal. Now we move into the organization of the spinal cord. As we all know, the spinal cord is divided into 31 segments: 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral, and 1 coccygeal. In a cross section, the spinal cord, as we said, it exhibits butterfly shape, which represents the gray matter of the spinal cord, which is surrounded by the white matter. حكينا في السباينال في الجراي matter only we can find the pericardium أو the cell body of the neurons. In the white matter we can't find any neuronal cell body, only the processes, which means axons and dendrites. The neuronal cell bodies within the gray matter, which has an interrelated functions, they are called nuclei. While within the white matter, they are called the group of axons which has an interrelated function, they are called tracts. Within the gray matter itself, we have the dorsal root, which extends posteriorly, and the ventral root anteriorly. The dorsal root, as we can see, they have afferent neurons, while within the ventral root we have the efferent neurons. The afferent neurons, they provide sensation from the targeted tissue, this, for example, the skin, and this sensation transmitted via the axons towards the sensory nerve cell body, which is present within the dorsal root ganglia. Those are the cell bodies of the dorsal root ganglia. Which type of neurons? They are pseudo-unipolar neurons. Then the stimulation goes to the central nervous system through the dorsal root. This is the afferent neuron with A. While the efferent neuron, usually those neurons have their cell body within the ventral root, and they send their stimulation towards the targeted tissue, for example, the muscle. They provide the efferent neuron, they provide an order to the muscle for contraction. Both the efferent and efferent neurons, they merge into a peripheral nerve. So the peripheral nerve contains both sensory and motor fiber. In this slide, this is a live section within a spinal cord. This is the gray matter, and this is the white matter. This is the dorsal root, and this is the ventral root or horn. Usually, the efferent fibers, they comes from the targeted tissue sensory towards their cell body, which is present within the dorsal root ganglia. This is the dorsal root ganglia. This bulging is the dorsal root ganglia, which contains the sensory neuronal cell body. Which type of neurons? Unipolar neuron or pseudo unipolar neuron. Then the stimulation goes towards the dorsal root of the gray matter. On the other hand, the efferent or the motor fibers, their cell body are present within the ventral horn of the gray matter. They send their stimulation or their order towards the targeted tissue. طبعا each segment within the spinal cord, whether it is lumbar or coccygeal or cervical, it has its own targeted tissue or muscle. So the efferent fibers merge with the afferent fibers to provide the spinal nerve or the peripheral nerve or the nerve trunk. So the peripheral nerve usually contains both types of fibers, the efferent and the afferent fibers, or the sensory and the motor fibers. In this section, it is light microscope section. We can see this is the butterfly shape of the gray matter, and within the gray matter we have the neuronal cell body. Within this white matter we do not have neuronal cell body, only the processes of the neuron. This is the dorsal horn, and this is the ventral horn. Dorsal horn, sensory, while the ventral is the motor neurons, where we have the neurons of the motor fibers. And within the ventral, dorsal fibers goes towards the dorsal root ganglia, which is located here, where we have the unipolar neuron. In this diagram, we can see the gray matter, butterfly shape, and the white matter. In the white matter, we can see only nerve fibers, axons, and their myelin sheath with the supporting cells, neuroglial cells. While within the gray matter, in this area, we have the neuronal cell body, the pericardium, supporting cells, glial cells, and the neuropil, or the processes of the neurons. As we said, motor neurons, they extend from the ventral anterior root. They provide the efferent neurons or the motor neurons, while the sensory neurons comes from the dorsal root ganglia, where they receive their signal from the targeted tissue, from the peripheral tissue towards the ganglia, then towards the dorsal root of the spinal cord within the gray matter. Generally speaking, the peripheral nerves are of three types, and this classification depends on their width or velocity. For example, type A fiber, usually they are myelinated fibers, diameter 4 to 20 micron in diameter. They conduct impulses at a velocity of 15 to 120 meters per second. Example are the skeletal muscle and the sensory fibers. Those are the examples of type A fibers. The type B fibers, less diameter of course, with less conducting velocity. They are of 1 to 4 micrometer in diameter, velocity of conduction 3 to 14 meter per second. Example are preganglionic autonomic fibers. The final type is the type C fibers. Those types are unlike the two other types. They are unmyelinated fiber. So we expect less diameter and less conducting velocity. Their diameter 0.2 to 1 micron. Velocity of conduction 0.2 to 2 meter per second. An example are the autonomic and sensory fibers within the nervous system. So this classification depends on the diameter, conduction velocity, and myelination of those fibers. All those three types of fibers are of the peripheral nerves, not the central, within the central nervous system. Now let's move to the nerve ganglia. What we mean by nerve ganglia? Ganglia means aggregation of neuronal cell body outside the central nervous system, or the locations where we can see the neuron pericardia outside the central nervous system. Those are classified into two types depending on their morphology and function. One of them is the spinal sensory ganglia or the dorsal root ganglia, which we explained earlier, and the autonomic ganglia, the second type, which is either sympathetic and or parasympathetic. Histologically, the spinal ganglia, it is found in the dorsal root of the spinal nerve, and it carries afferent sensory impulses. Histologically, it is surrounded by a thick connective tissue capsule. It surrounds the whole ganglia, and usually the type of neurons which are present within those ganglia are pseudo-unipolar neurons. They are not multipolar neurons. They are pseudo-unipolar neurons or unipolar neurons. They are only the neuronal cell body. They have only trophic functions, and they do not receive synapses. And we can see lots of satellite cells surrounding those pericardia. So we have a high intensity of satellite cells surrounding the pericardia. Usually, neuronal cell bodies of the dorsal root ganglia, they are aggregated towards the periphery of this ganglia. It is not distributed all through the ganglia. We can see them only within the periphery of the ganglia. As we can see in the middle, can't see any of those neuronal cell bodies. They are not distributed all through the ganglia. Instead, they are located within the periphery of the ganglia. And the ganglia usually is surrounded by a thick connective tissue capsule. Again, they are distributed on the periphery of the ganglia. They do not form synapses, so the neuronal cell body have atrophic function. They are pseudo-unipolar neuron, and the ganglia all through surrounded by a connective tissue capsule. The second type is the autonomic ganglia, whether it is sympathetic or parasympathetic. Usually those type of ganglia, they do not have a connective tissue capsule. Occasionally, we may see a thin connective tissue capsule surrounding this autonomic ganglia. Most of those autonomic ganglia are intramural. What we mean by intramural, they are within the organ itself, like the GI tract and the bladder, where we do not have capsules for those ganglia. Those intramural, we can see the pericardium of the neuron. What's the differences between the autonomic ganglia and the sensory ganglia? Within the autonomic ganglia, we have synaptic connection. Within the dorsal root ganglia, we said that the neuronal cell body only have trophic function. They do not form synapses. Within the autonomic ganglia, we have synapses. Also within the autonomic ganglia, those types, the neurons are multipolar neurons. Within the dorsal root ganglia, it was unipolar neurons. The intensity of satellite cells which surrounds the pericardium are fewer than the dorsal root ganglia. The fibers usually unmyelinated fibers of the autonomic ganglia, بينما في الدورسال root ganglia usually they are myelinated fibers. The final difference is that within the autonomic ganglia, those pericardia usually, or the neuronal cell body, or usually distributed all through the ganglia. Unlike the dorsal root ganglia, as we said, they are only present towards the periphery of the ganglia. Again, what are the differences between the autonomic and the dorsal root ganglia? First of all, autonomic ganglia usually do not have capsule. Some occasions it has a thin connective tissue capsule. Within the autonomic ganglia, we can see synapses surrounding the neuronal cell body. Within the dorsal root ganglia, there are no synapses. Neurons within the autonomic ganglia are multipolar neurons. Within the sensory ganglia or the dorsal root ganglia, they are unipolar neurons. We have fewer satellite cells. On the other hand, within the dorsal root ganglia, we have lots of satellite cells surrounding the pericardium. The neuronal cell bodies within the autonomic ganglia, they are fairly distributed all through the ganglia. On the other hand, within the dorsal root ganglia, they are only gathered within the periphery of the ganglia. The axons within the autonomic ganglia, usually they are non-myelinated axons. On the other hand, within the dorsal root ganglia, they are myelinated axons. Those are generally the main differences between the two types of ganglia. Again, ganglia means aggregation of neuronal cell body outside the central nervous system. Now we move to the organization of a brain. The organization of a brain, or cerebellum and cerebellum, cerebrum and cerebellum. Though this organization طبعا احنا رح ناخده in general. الديتيلز رح تاخدوهم في central nervous system module. This organization, the importance of it, that we have the gray matter is from outside, unlike the spinal cord where we have the gray matter in the middle of the white matter. For the brain, the gray matter is from outside, and the gray matter forms two parts, the cortex, or it forms the cortex, and the white matters form the inner medulla of the brain. طبعا within the gray matter also we can find the neuronal cell bodies. Still within the medulla we can find the neuronal cell bodies. In both we can find neuropils and the neuroglial cells. This is what we need to know about the brain. Cerebrum. Now we move to respond to neuronal injury. The neuronal injury, as we said earlier, that the neurons are post-mitotic cells. For that they can't divide, so the regeneration ability of the neurons are very, very limited. So the damage or an injury to the neuron could occur either at the neuron cell body itself or at one of the processes, mainly the axon. Our concern is the axon. The damage to the cell body. Once we say a damage to the cell body, pericardium, this is the big one. If the damage occurs on the pericardium, there will be a degeneration of the neuron, the damaged neuron, also an atrophy for the targeted cell or the skeletal muscle of this neuron innervating the muscle. Also, a third degeneration occurs if other neuron forms a synapses with the neuron. So if we have a neuron which forms the synapses with this neuron which forms the synapses with the injured neuron will cause a degeneration of the synaptic neuron. This is what we call a transneural degeneration. Those three degenerations occur if the damage within the cell body. On the other hand, if the damage at the axon, there are two types of processes. Initially we have a degeneration which takes place almost 2 weeks, then a regeneration will take place. So if the damage to the axon, initially we have a degeneration, then a regeneration takes place. As we can see, this is the injured side. There should be initially a degeneration. The degeneration towards the distal part of the neuron is called anterograde degeneration, which involves the distal part of the neuron towards the targeted tissue. This part will completely degenerate. This anterograde degeneration is called Valerian degeneration, which involves the complete degeneration of the distal damaged part of the neuron, and this type of degeneration takes 2 to 3 days to complete, and this will get involved by the phagocytic ability of the microglial cells, Schwann cells, and the monocytes to engulf the fragmented, damaged tissue. Also, we have a retrograde degeneration. This retrograde involves the proximal almost two internodal segments. Those two internodal segments usually degenerates. Meanwhile, during this degeneration, the distal and the proximal, as we can see, the cell body itself has its own changes. As we can see, the nucleus goes eccentric towards the periphery, the cell bodies disappears, or cell body, what we called, if you recall, cell body reaction or chromatolysis, which indicates the low activity of the cell, swelling, eccentric nucleus, and chromatolysis or cell reaction. As we can see, complete degeneration of the distal part, which means the Valerian degeneration. Then the regeneration will take place. The regeneration usually starts at the 3rd week. Within the third regeneration, initially, pericardium regains its activity and its shape. Cell body appears again, which indicates the activity of the cells. And as we can see, the Schwann cells starts to form bridges, and it starts to divide to form bridges within the damaged sites. Meanwhile, the axon nerve fiber will start to grow, forming sprouts or neurites. Those neurites, many neurites will form. Not all of these neurites will be successful to gain the original shape of the nerve fiber. The only successful one will regain the original shape. The growth of those neurites usually 3 millimeter per days. Once those neurites regain the original shape of the fiber, then a complete regeneration takes place within the axon. Then at 3rd week, again, bridging, dividing of Schwann cell, neurites forms, growth of the fibers by sprouts. Some of those neurites will regain the original form towards the targeted tissue. Some of them will degenerate. If those neurites fail to regain the original form due to the long distance of the injury or whatever the reason, then they will go into the form what we called neuroma, and this neuroma will cause death of the targeted tissue.